And I'll just hand it off to the first tutor.
Hi, okay, I'm gonna share my screen. All right, so today we're going to be talking about galvanic cells or electrochemical cells, voltage and standard reduction potentials. And all of the information in these slides will be either from the textbook or from the lecture notes if you want to reference it. So I guess the first question you have to ask is like, what is an electrochemical cell? So basically it's just a battery that's powered by connecting two redox reactions into a circuit. So in order to understand this, we're going to talk more simply about just thinking about two molecules, A and B. And so A donates electrons to B spontaneously, becoming oxidized as B becomes reduced. This means that the oxidized form of A is lower in free energy than the oxidized form of B, as the reaction is occurring spontaneously. So because it happens spontaneously, we know that the oxidized form of A must be lower in free energy. Because an electron transfer is involved, we can easily relate the change in free energy of this equation to the voltage of an electrochemical cell containing these two molecules. So now we're going to talk about a more realistic situation with metals. So to understand this, we'll look at a specific case in which there's a piece of zinc metal placed in an aqueous solution of copper sulfate. As seen here. And so the metal rod is known as an electrode, and an electrode is a metallic conductor that is in contact with solutions or other non-metallic parts of electrical circuits. So when like this happens, once it's placed in the copper sulfate, the zinc electron will dissolve and the copper metal will get deposited onto the zinc electrode. And this is seen in these equations with the solid turning into the zinc in solution and the copper in the solution turning into the solid. So it's kind of like a standard redox reaction that you would have seen in like a Chem 1A class. So this reaction is spontaneous with these two metals. However, if you do the opposite, putting copper electrode in a zinc sulfide solution, the reaction isn't spontaneous. So just as we concluded in the example using like the simplified version of just having A and B, we can conclude that the copper metal therefore has a lower free energy than the zinc metal, and that's why it occurs spontaneously. So in order to construct a working electrochemical cell, we need to cause a current to flow as a direct result of the spontaneous nature of the two half reactions. And so we're using the zinc solid turning to solution and the copper solution turning to solid because those were like the spontaneous relationship that we observed before. So now, before we go over exactly how to make like the standard electrochemical cell, I want to look at this picture on the left and what do you guys think will occur in the image to the left? And so to help understand it, it's a zinc electron, which is the silver, placed in a solution of both copper sulfate and zinc sulfate. And this is also a copper electrode. So take a minute or two to think about this, and then let me see if you can talk or just send something in the chat. So yes, so the zinc electrode will dissolve and give off two electrons, but those electrons won't flow through the wire, as someone said in the chat. There's enough copper 2+ ions in the aqueous solution that the electrons will just reduce these Cu2+ ions and they won't go through the circuit because it's just easier they'll be coming in immediately into contact with copper 2+ ions in the solution. So no current will flow through the wire. And now, so this is another possible situation. And this is, again, it's the zinc electrode in solution and the copper electrode in solution. But again, they're separated, but why won't current flow? You can like take a few minutes to think about it and then send it in the chat again. Okay, so yeah, the zinc atoms will dissolve, releasing 2 electrons, but there's no copper ions in the solution, so the electrons can flow to the other cell through the wire, which addresses the first problem of the previous cell, but this would lead to a buildup of positive charge in the left cell, and so no current will be sustained. And so now this is an actual circuit that will work. So the way to complete this circuit is you can't make a completely permeable membrane because then you'll just be back at the first situation in which the electrons from zinc will just immediately reduce the copper 2+ in solution. So you need to have a semi-permeable bridge between the 2 electrodes that impedes the flow of copper and zinc ions, but allows the flow of sulfate ions. So there won't be a charge buildup, which addresses the problem in the previous slide. So how this will work is that zinc ions will release electrons at the left electrode, and these electrons will move along the wire conductor until they reach the copper electrode, where they'll react with the copper ions. And then eventually the sulfate can move from the right to the left to address the charge buildup. And so the way that these are defined is the anode is the negative electrode or the source of the electrons, which in this case will be zinc. And then the cathode is the positive electrode, or in this case, the copper. And so this is the conventional way that you would like write these if you were doing like the shorter version. Make sure to note like when you include like the charges, when you include the state, and like how many lines go in between each. So now we're going to talk about the standard cell potential, which is like a characteristic of the electrochemical cells. So the driving force of the current flow is given by the differences in free energy between reactants and products, and this is dependent on concentration. So as the reaction progresses, the concentration will change and the current will change with it. And eventually the system will reach equilibrium and the current will stop. So the voltage difference between the 2 electrodes will also change with the concentration. And we know then that the voltage generated by the cell depends on the concentration of the reactants in each half cell. So the standard cell potential is defined as the voltage generated by an electrochemical cell in which the reactants are all at standard concentration. And also I think it's 298 degrees Kelvin. So this is just a figure of how you would actually measure the voltage. It's not like as important, but if you would want to know like the way in which you do it, you have an electrochemical cell connected to an external power supply that puts out a constant voltage, and the circuit is completed with a resistor. And so then the external voltage can be matched to the voltage of the cell, and it will give you a reading on like the voltage of the cell. So another thing that's really important in this class, a lot of things end up relating to free energy change and like equilibrium constants. And so standard cell potential is no exception. So it's important to recognize that standard cell potential and standard free energy change are fundamentally related. So to understand this, we need to recognize that the voltage generated by the cell is related to the electrical work that can be done by the cell. And as we learned before, electrical work is powered by the underlying chemical reaction, and the free energy change of that reaction will give the maximum amount of work and be extracted from the reaction. So in order to relate cell potential and free energy, we need to first calculate the amount of work that can be done by the cell under standard conditions. So in order to understand this, we just need to briefly review electrical fields and electrical work is probably something that you would have learned in like a physics class, but it's good to refresh on it before going further into this material. So consider an electric field generated by, for example, two metal plates. And then this is the equation for the force experienced by a charged particle within this electric field. So the F is the force, the Q is the charge of the particle, and E is the electric field. So if we want to move this particle some small distance, then the work done on the charged particle will be equal to, or dW equals negative F dr equals negative QE dr. So just substituting in the equations. And this work is going to be negative because it's being done opposite to the electrical force. And the work being done will therefore change the potential energy of the particle, and it'll be at the same magnitude. So it'll be negative QE times dr. Putting this in finite terms, or like moving like a specific distance, the change in this will give you a change in potential energy equal to Q times delta E, where delta U is the change in potential energy, Q is the charge of the particle, and delta E is the electric potential difference, or like the voltage. So that was just a review of the physics, but going back to electrochemical cells, the electric potential of an electrode is labeled as E, and with the left being EL and the right being ER. And so the voltage generated by the cell is going to be delta E, which will equal to delta E right minus E left. So it's just the change between the two. And we also remember that electrons will flow from regions of low electric potential to regions of high electric potential. So if delta E is positive, that means that electrons are actually moving from the left cell to the right cell. This can get a little confusing because oftentimes when we're talking about stuff like this, things will move from a region of like high potential to low potential, but because electrons are negative, it's the opposite way. And so as given by, or at standard conditions, the standard voltage will be given by this equation, and where the ER naught and EL naught are just the standard electrode potentials. So now we're going to go back and relate the previous two concepts, so relating cell potentials to the free energy change. For this electrical cell, work is going to be equal to charge moved times voltage, which was explained in one of the previous slides with the Q times delta E. And so for a reaction in which V moles of electrons are transferred, work equals negative V F delta E, where F is the magnitude of the charge on 1 mole of electrons, and delta E is the voltage of the cell. So this last slide is mainly just like math equations, but the important thing to know is that this is the maximum amount of work that can be extracted from the cell, which can be equated to the free energy change of the reaction. So this should also hold at standard conditions. And then if we use the relationship between free energy change and equilibrium constant, we can substitute that into the equation, which will give us a direct relationship between electric potential and equilibrium constant. So looking sort of at like the voltage of a cell can give you information about how far the reaction has progressed. That's the presentation. Does anyone have any questions? If not, I'll just pass it on to the next tutor then. Thank you.
Okay, hi everyone. I'm just going to share my screen. Can everyone see okay?
Yeah.
Oh, thank you. So I'm going to be talking about the Nernst equation, which Heather just touched on briefly at the end of her slides. So first, what is the Nernst equation? So the Nernst equation is an equation that relates the reduction potential of an electrochemical reaction to the standard electrode potential, temperature and concentration of chemical species undergoing the redox reaction. So kind of give you an intuitive feeling for what the Nernst equation is. if you think about the free energy equation, right, you have delta S minus delta H minus T delta S. So that kind of gives you that has two components, right? That has an energetic component, that's your delta H, and then you have your entropic component, your T delta S. So in your standard chemical reaction, right, the ones you guys are comfortable with, delta H kind of captures bond breakage and bond formation, right? But in a lot of redox reactions, your electrons just move from one atom to another. There's no bond breakage or formation, even though there can be, and a lot of times there isn't. So how is it that we can capture the energy change or the energetic component of the free energy associated with this electron movement? And that's where the Nernst equation comes in. So we have to try to relate the movement and energy of electrons to free energy, to our free energy equation. And here is just an example equation, example question of what we can answer with the Nernst equation. And right now, you guys, if without this presentation, I don't assume that you guys can answer this, but this just gives you an idea of what kind of question we can answer with this. So given that the standard electric potential that Heather just talked about is a certain voltage for a certain reaction, what is the equilibrium constant for this reaction, right? So we have this standard reduction potential, which characterizes the spontaneity of this reaction. And we want to know when the reaction, there's no net movement in the reaction, what does it look like? So this is the kind of question we want to answer. So first, if we want to derive the Nernst equation, it's a lot simpler than the other derivations that you guys have had to learn. So first, we just start with our free energy equation, right? So this, you guys have seen this before. And then the critical part is we have to relate free energy to the potential difference, the voltage, right? So just to clear up some terms in case you guys are confused, voltage, potential difference, this E, V, they're all the same thing. It's just the difference between two potentials. So if you look at this, this might not seem intuitive, right? For now, just ignore the negative sign. I'll get to the negative sign in a moment, but I just want to convince you that this term is an energy term, right? So N is moles of electron, and F is Faraday's constant, which is charge per mole of electron. So we multiply these two things together, we just get a charge. And E is your voltage difference. So in case you guys didn't take the physics class that talks about voltage and potential, I'll just go into it a little bit. So voltage, we separate potential and potential energy because potential energy, if you remember from the previous portion of class, right, your equation is your Coulomb's constant times one charge times the other charge divided by the distance. But that depends on two charge. It's a lot easier for us to calculate things if we just depend on one charge, right? So in every case where there's two charges, we can view one charge as the source charge and one charge as the test charge, what we place into the potential generated by the source charge. So the voltage is just the potential calculated from the source charge. And I'll get a little bit more into this on the next slide. But the idea is this is just charge. This is just the potential difference between two states. And if you multiply your charge by your potential difference, you get an energy term. And this is how we equate a potential difference to your free energy. So why is there a negative sign, right? This is what confused me a lot. So I kind of want to spend just a little bit of time so you guys don't get confused. So Heather mentioned that electrons move up their potential, right? So we used to free energy move down, right? A negative delta G is spontaneous, but a positive standard reduction is spontaneous for electrons. So why is this? So in the corner, you see V equals KQ over R, right? Where K is Coulomb's constant, Q is your source charge that I was talking about, and R is the distance. So kind of going back to the idea of a potential. So you have a source charge, you place it in space, and that generates a potential around it. At every point in space around that source charge is this quantity called a potential. If you multiply that potential by the charge of the test charge that you place in that position, you get the potential energy of those two charges in that position. So let's look at what happens. Imagine what happens if we have a positive source charge and a negative test charge, right? If we have a positive source charge, the voltage is positive at this position, right? Or at all positions around it, because Q is positive, K is positive, R is positive, the whole thing is positive. But positive charges and negative charges attract, right? So if I place an electron into a potential generated by a positive source charge, R decreases, right? So if R decreases, V increases. Do you guys see that? So if the denominator gets smaller, then the whole thing gets bigger. So when we have the case of a positive source charge and a negative test charge, like an electron, the electron moves towards higher potential, higher V. Now let's look at the case where we have a negative source charge. If I have a negative source charge, the potential everywhere is negative. If I place an electron, another negative charge, into the potential generated by the negative source charge, the two negative charges repel, right? So R increases. But since your potential is already negative, as R increases, it approaches zero. So going from a negative number to zero also increases potential. So we see that in either case, whether it's a positive source charge, whether it's a negative source charge, if I place an electron into that potential, the electron likes to move towards higher potential. So this is why we have to put that negative charge. So we know that a spontaneous movement of electrons is associated with a positive change in potential. So positive delta V, positive E, it's the same thing. But we also know that reactions like to have negative delta G. That's what we know is spontaneous. So in order to make those two signs consistent, we put a negative sign to make the positive spontaneous voltage difference matching the negative sign of the spontaneous free energy change. Does that make sense? Does anyone have any questions? If not, I'll just move on. And then the rest of the derivation is easy, right? So we just had our delta G equals delta G standard plus this term, and now we just substitute into the expression this NFE term. And then on one side, it's just your free energy, and then the other side is your standard free energy. So we just substitute in these standard terms. And then if you divide by the charge, so divide by a number of moles of electrons times the charge per mole of electrons, you get this term. And this is the Nernst equation. Does anyone have any questions about the derivation? Okay, if not, I'll continue. So here, I think I found a useful diagram to kind of relate free energy and your standard reduction potential. So this side of the triangle is what you guys have been dealing with in class, right? So if I give you a standard free energy of a reaction, you can give me the equilibrium constant and vice versa. But now that we've kind of incorporated the standard reduction potential of a cell, but it's not always cell, right? Sometimes you can characterize biological systems as a quote unquote cell. But in any case, if I give you that standard reduction potential, you can calculate the free energy change, the standard free energy change, calculate the equilibrium constant. All of these are interchangeable. If I give you one, you can calculate the other two. So this is how they're kind of related. And now let's revisit our first example, right? So if we have a standard potential of a cell is 1.1 volts for the zinc copper redox reaction, the reaction's written there. What is the equilibrium constant for this reversible reaction? And I haven't shown you, I haven't done the, I didn't write the calculation in the PowerPoint. I kind of want you to think about what is the condition that allows us to find the equilibrium constant, right? So equilibrium constant, instead of this Q, this reaction quotient, we just have K. So what is the constraint that allows us to find K? So equilibrium constant is when the free energy is zero, right? When there's no net reaction anymore. It looks the same. And we just learned that electrons like to move up their potential, right? So it stands to reason that once there is no more potential difference, electrons will not move. There will be no net movement of electrons. If between 2 points in space or 2 states, there's no voltage difference, there's no potential difference, then the electrons will move. So if we set this potential, this E over here, equal to 0, then you can just solve for K here. And that's how you find your equilibrium constant. Does that make sense to everyone? Does anyone have any questions? Okay, if not, I'll continue. And then here, this is my last slide. I just kind of wanted to give you some food for thought. So in the examples that Heather gave, the cells you guys have been using are of different metals, right? But that's not necessary to generate a cell with a workable voltage. If we look at the Nernst equation, here, if we have two cells, right, and two cells, and each electrode uses electrolytes of the same metal, we have a standard reduction potential of 0, right? Because the reactions happening at one electrode are exactly the same as the reactions happening at the other electrode, right? So one is just an oxidation, one is a reduction. But the metal being oxidized and reduced is exactly the same. The reaction is exactly the same. So the standard reduction potential is 0 because they just cancel out. But we still have this term here, right? ln of this reaction quotient. So if we change this reaction quotient to be less than 0, then this term will be negative. Multiply this negative is positive. We get a positive voltage. So we want the denominator in our reaction quotient to be larger than our numerator. And this is kind of the basis of the concentration cell. You don't need to use two metals. You can just use electrodes with different concentrations of electrolytes. So kind of think about which, if we have two electrodes with different concentrations of the same electrolyte, which electrode's cathode, which one's anode, right? So does the cathode have the higher concentration of electrolyte, or does the anode have a higher concentration of electrolyte? You guys can think about this, and I will put the answer in the worksheet. Well, that's it. Thank you, everyone. Does anyone have any questions? If not, I will stop sharing and hand it off to the next tutor.
Hello. Okay, I'll be talking about redox reactions and electron carriers. And one type is, oh, wait. Okay, never mind, sorry, I thought I was muted. So one type of biologically important redox active metals are those bound to proteins. And you're going to have these coordinated metals. Here I've represented a protein. These can be redox couples. So if one is reduced, so give electrons to this one, then this one will be oxidized. These are important in photosynthesis and oxidative phosphorylation. An example of this would be a heme group, which is a porphyrin ring and iron. And you can see this in cytochrome C, which is important in oxidative phosphorylation. So the heme plus the protein is the cytochrome. So here we have the porphyrin ring, and the periphery attaches to the protein. So the squiggles are the protein. And if you flip the ring perpendicular to the plane of the paper, you'll see the ring and the iron, and it's the iron is coordinated by two of the protein side chains. Here you have a histidine, and then here you have a methionine. Another example you'll see also in the electron transport chain are iron sulfur clusters, where the iron is coordinated with the sulfur atoms of cysteines and methionines. And yeah, so another prosthetic group in the electron transport chain. And the thing about these is that they're one electron carriers, right? So if you have iron with a positive charge and then it gets one electron, then you're going to get iron with a +2 charge. Okay. The next group of carriers are nicotinamide adenine dinucleotide, or NAD+. And you can see how it's dinucleotide because you have one nucleotide right here and another nucleotide right here. So this is the adenine monophosphate and the nicotinamide mononucleotide. And this is the redox active part. So if you, and this is the oxidized state. And so if you add 2 electrons and a proton, which is a hydride ion, then you reduce it, and then you see that now you don't have a positive charge anymore, and you have another hydrogen on the ring, right? So here's our net equation down here. You can, and so this is a hydride ion. And you can see on this if you add a phosphate onto this oxygen right here, it's NADP plus, and similarly NADPH. And the phosphate isn't involved in the redox, but it's important for the cell because you need these two types of redox active molecules. So you have two different pools, and they're used for different functions. So NAD will be used in catabolic reactions, so TCA cycle, things like that. And then NADPH, you'll see it in photosynthesis. It has antioxidant functions. So it's important in anabolic reactions. For example, you need NADPH to make carbon sources if you're a plant. I mean, you're not plants, but anyway, okay. So NAD is 2 electrons at once because you have the hydride ion. For an example of the mechanism, we can look at alcohol dehydrogenase. And so I have ethanol here, and when you oxidize it, it becomes an aldehyde. And you can also go in the reverse direction. So here we have NAD plus, and then we're starting with the alcohol. So let's say that in the alcohol dehydrogenase, there's a base that can abstract this proton here, and then you can imagine the 2 hydrogens here. And so the hydride goes on to the NAD plus, and then you wind up with an extra hydrogen over here and an aldehyde. So yeah, this is the redox active part of NADH. Also the extra proton from here, okay? Okay. Another class are the flavins and quinones, which can be oxidized or reduced in 2 steps of 1 electron each. So it's sequential. And these are vitamin B2 derivatives, and the sequential manner of the reductions or oxidations helps so that you can couple processes that transfer single electrons to reductions of NAD plus, which requires 2 electrons. So first we'll look at the flavins. This is riboflavin, and if you have an R group of adenosine diphosphate, then you have flavin adenine dinucleotide, which you know as FAD. So if you add 1 electron and 1 proton, then here you have the radical, and then you can think about it like this bond swinging out to capture the proton, and another electron and another proton, and then see, now you have another hydrogen here, and then you have the formation of a double bond here. These flavins are always tightly bound to proteins. An example you might know is complex 2 of the electron transport chain, whereas NAD plus and NADP plus are soluble, so they're floating around in the cytoplasm. And that's because you can imagine it looks like a nucleotide, and your nucleotides are also floating around in the cytosol. Another electron carrier are the quinones. This R group is often a hydrocarbon tail, so an aliphatic R group makes it so that it's membrane soluble in all 3 of these forms, and so it's going to localize to the membrane, and it can act as a mobile electron carrier in the membrane for complexes that might not be able to move around as easily. So here, electron, proton again, and you have a radical, and you get an alcohol group, and then electron, proton, and now you have 2 alcohols. Okay. Any questions on any of that? You've probably seen a lot of these things in your other classes. So these electron transfers are important in how we make ATP from the food we eat. And oxidation of glucose releases a lot of free energy. So yeah, this is a lot of free energy. And if you split this equation into two half reactions, the oxidation half will be this, and then the reduction will be this. And so you can see oxygen to water is a reduction, and you have the movement of 24 electrons. So in metabolism, these electrons are transferred to NAD plus and FAD. In glycolysis, right, produce 2 NADH from 1 glucose, and then you can take these 2 pyruvate, make more NADH, and then take your acetyl-CoA and take them through the cycle, which you probably all have experienced in other classes as well. And the free energy stored in NADH and FADH2 is used to generate the proton gradient across the mitochondrial membrane. And this stepwise oxidation is important as opposed to just combusting the glucose, which would give you a lot of heat, an explosion, and that can damage your cells. And you'll lose a lot of the energy as heat, but the stepwise oxidation is a lot more efficient than oxidation in one step. Your cells can harness about 50% of this free energy from glucose and convert it to ATP. So as you oxidize glucose in a stepwise manner, you release a little bit of energy at a time, which is used to reduce NAD plus and FAD, and then you can oxidize those to make ATP. Yes. So any questions there? Okay. Well, I think there's a worksheet question on the ratios of NADH and NAD plus and NADPH and NADP plus in the worksheet. So just it's a thought question. So yeah, if you want to see the answer, go see that later, and I will pass it on to the next tutor.
Okay, let me just share my screen. All right, so what I'll be examining is a little bit of a closer look into the redox of cellular respiration, and I'll also be touching on photosynthesis a little bit as well. So first, we can view cellular respiration as a redox reaction, as Ryan introduced. So of course, this is the equation, chemical equation for glucose being combined with oxygen and undergoing combustion to form water and carbon dioxide. So the first thing I wanted to illustrate, just to get a sense of what exactly is being reduced and oxidized, is to introduce some of these oxidation rules. So you've probably seen these from Chem 1A or whatever your general chemistry course was, but just sort of as a refresher, these are how the rules work. And I think just to give everyone an example calculation of how you would assign an oxidation state, I can lead you through the example of the glucose molecule, which I have pictured right here. So essentially, the idea behind assigning the oxidation numbers is you want to count the number of valence electrons an atom normally has, and subtract from that the number of electrons that that atom kind of has for itself. And what I mean by that is you have to examine all the bonds that the atom is participating in. So for example, if the atom is bonded to an atom that is more electronegative than itself, then it doesn't really have those electrons. But if it's bonded to an atom that is less electronegative than itself, then it has both of the electrons from that bond. If it has a lone pair, both of those electrons become it. So in this example, we can see that if you consider all of the hydrogens in the glucose molecule, they're either all bonded to oxygen or a carbon. And both of those atoms are more electronegative than hydrogen itself. And so for that reason, hydrogen doesn't really have any electrons of its own, and hydrogen normally would have one valence electron, so 1 minus 0 is positive 1. And then just one other example that I can do is if we consider oxygens in this molecule, you'll see that the oxygen is typically bonded to two carbons, and also because it's the most electronegative atom in this entire structure, essentially it retains all the electrons from its bonds. So then the calculation would be 6 minus 8, which is negative 2. However, if you look at the just diatomic oxygen molecule, you'll notice that it has an oxidation state of 0 because it's essentially the same molecule. And so as a result of this, what you can see is that as this reaction progresses, you go from having a carbon that has an oxidation state of, on average, +0 to now an oxidation state of +4, and then your oxygens become -2 from your diatomic oxygen of 0. And so what's really happening is that your carbon is being oxidized while your oxygens are being reduced when they're converted into water. And then the other observation that I would add on to this is that photosynthesis is simply just the reverse of this reaction. It quite literally is the reverse. So you can kind of take the inverse of what is being reduced and oxidized. And so next, we can discuss the idea of the stepwise reduction and oxidation that happens in biological systems. So at the end point of cellular respiration, you of course have the electron transport chain. And the way that energy is produced there is you have these protons that are coming from the outer membrane space of the mitochondria, coming through the F1 ATP synthase and combining with oxygen to make water. And so normally if you've mixed hydrogen and oxygen together, you'll know that it is a pretty explosive reaction. And so it can be pretty difficult to harness all this energy at once. So if you could think back to your last unit, think about a cylinder either expanding slowly or a cylinder expanding all at once. So if a cylinder expands rapidly and all at once, it becomes very hard to control that and to harness any energy from it. And so what your cells do is go through a number of very slow processes in order to extract the most amount of energy from it. And so typically what happens here is that you have a series of enzymatic reactions that will just move electrons from low potential to higher potential. And to give you a concrete example of what I mean by that, if you consider a lot of the hydrogens in the glucose molecule, they're typically, most of them are bonded to carbons. And so when a hydrogen is bonded to a carbon, it's at a relatively low potential. And so you can think of an electron at low potential as somewhere on here in this hypothetical imaginary electric field. And as the carbon gets further oxidized, the proton eventually has nowhere to go and eventually ends up on the oxygen where it's at the highest potential. And so then you can think of it as here. So this is just a distinction that I would make, that although at every step of this process, you're going into more and more negative for free energy, you're going from lower potential to higher potential. And this is favorable for electrons because you can kind of think of this as, if this electron is negatively charged, then this association with something that's positive, the closer it is, the more favorable that will be. So does anyone have any questions on this so far? Okay, I will proceed. And so now just to give a quick recap of what happens in cellular respiration, I'm sure many of you have seen this before, but essentially the most important intermediates in this process are your NADH and your FADH2. And so just to point out, like I was discussing previously, essentially you can kind of view the entire cycle of cellular respiration as further and further oxidizing some carbon-containing molecule. And of course, these carbon-containing molecules are refreshed because you're adding more and more glucose through the cycle. And so as you continuously oxidize the carbons on this molecule, you either release them completely as CO2, because this is kind of the farthest that a carbon atom can be oxidized, or you use them to generate NADH by reducing the NAD positive electron carrier. And these become pretty important intermediates, as I'm sure you've all seen. And that leads us to the electron transport chain. So I'm just going to put some text up here, but let me explain what I mean by all of this. So for the purpose of this class, I think you don't need to know too many of the specifics as to what exactly each one of these molecules are. And also we make the simplifying assumption that for every single NADH molecule that's added to the electron transport chain, approximately 3 ATP are produced. And for FADH2, it's a little bit less than that. And to kind of go over how exactly NADH translate into number of ATP produced, the exact process of this is that these electron carriers interact with the complexes of the electron transport chain. And as a result of these electrons moving in a way that minimizes their free energy, so again, for an electron to minimize its free energy, it's moving from a low potential to a higher potential because electrons are more attracted to more positively charged things. And as a result of this movement, you have pumping of hydrogen protons across the membrane, and these protons are ultimately what causes the generation of ATP. Furthermore, you'll note that FADH2 actually makes fewer ATP, per electron carrier than NADH. And the reason for that is that it just enters the electron transport chain later. And so the electrons it adds are able to pump fewer protons across the membrane, and that's why fewer ATP are produced. In reality, I think in some of your classes, you might have seen that one NADH molecule actually makes 2.5 ATP instead of 3. And the reason for this is that in some cases, because some of your NADH are actually generated during pyruvate oxidation or during glycolysis, and those things have to be shuttled from outside of the mitochondria to the inside, that uses up ATP. But for the purpose of this class, you can just consider 3 ATP as the number that's produced per NADH molecule. And for FADH2, we don't really discuss it, but I don't think you will need to know it. And so next, what's mentioned in lecture is a calculation of the efficiency of cellular respiration in terms of producing ATP. So let's think about how we would go about this calculation. So we know that for every single glucose molecule, you can generate negative 2,800 approximately kilojoules if you just combust it directly. So since our cells are using glucose for the production of ATP, how efficient is this process? So we can make a few assumptions here. So we can assume that, first of all, that one ATP molecule, it requires approximately negative 30 kilojoules per mole, and also that one NADH molecule represents 3 ATP that are produced. And so what we can recognize from the process of cellular respiration is that essentially all the ATP are produced during the electron transport chain. And so really the relevant redox reaction is just going to be involving carriers such as NADH and also the reduction of O2 into H2O. And also you'll note that on this reduction table, it's given NADP plus instead of NADH, but actually if you look at the biochemical standard state reduction potential for NADP plus, it's actually the same as NAD plus. And so here, this is the overall reaction that's occurring, and then you can split this into your reduction reaction and your oxidation half reaction. Note that I've switched this to positive because I've reversed this reaction. And you can apply this formula that your delta G for biochemical standard state is given by this. And that leads you to a calculation for at least at biochemical standard state, what the free energy of converting, what the free energy of this redox reaction would be. And so if you further apply some of the assumptions that I've talked about, and this is mentioned in the lecture notes as well, this yields approximately a 41% efficiency. And so next, briefly, I want to talk about the redox that's involved in photosynthesis. So this is comes directly from your lecture slides, and this is something that I found that's representative of just where everything is, just to kind of help you get your bearings. So when we talk about the thylakoid space, so this area right here where the water splitting happens, that's the inside of the individual thylakoid stacks in the chloroplasts, and the stroma is essentially everything that's outside. So just notice that the NADP plus is getting reduced on the outside, whereas the water molecules are being split on the inside here. And the main thing to take away from this is that the first step of this reaction, which involves the oxidation of H2O back into O2, because as we remember, photosynthesis is literally just the reverse of cellular respiration. This is an extremely unfavorable reaction because oxygen is just such a strong oxidizing agent. So to oxidize it is very difficult. And that's why it's mentioned in lecture that this takes approximately 4 photons. And I believe that represents about 600 kilojoules of energy. And the end result of this is that this entire reaction requires about 200 kilojoules to drive it. And so the efficiency of this process is roughly 30%. And that's also mentioned in your lecture notes. But just the main point here is that this step is very unfavorable because of how strong of an oxidizing agent oxygen is. And so next I have the worksheet question, but does anyone have any questions about what we discussed so far? Okay, and I realize that I'm running a little bit low on time, so I'll just try to read this question and introduce some of the main ideas to think about, but here's a standard reduction potential table that might be helpful. So consider one of the steps in the TCA cycle, the oxidation of malate into oxyl acetate, coupled with the reduction of NADP plus to NADH. Is this reaction spontaneous at biochemical standard state? What does this say about the concentrations of these species inside the cell? And keep in mind that this reaction happens in the inner membrane of the mitochondria. So what do we know about the pH in this compartment? And what you can do if you work this out, and this will be posted in the worksheet solution as well. So if you work this out, what you will realize is that at biochemical standard state, this reaction is actually not spontaneous. It has a delta G standard of about 29.7 kilojoules. But from our own knowledge, we know that the TCA cycle actually progresses. So this means that this reaction does have to happen in vivo in order for our cells to work and for the rest of cellular respiration to work. And so what this means is that in order to drive this process, there have to be things that are essentially deviations from standard state. So the concentrations of the reactants, for example, might be very high. So that means inside the cell, the ratio between oxaloacetate and malate is pretty low because malate is the reactant here. And also by keeping a high quantity of NAD plus, the cell can essentially drive this reaction inside the mitochondria. And just one other main idea is that the pH inside the inner membrane space is a little bit higher since it has the low hydrogen concentration in order to drive the synthesis of ATP. So those are just the main takeaways from this question that I wanted to get across. So with that, I'm just going to stop sharing. Does anyone have any questions really quickly? No? Okay, so then with that, I'm going to end the recording for today.